Good afternoon, everyone. We'll start in 2 3 minutes. Sure, sure. Okay, let's start. So today's session is basically the summary of all the sessions. I'll be going through some questions, what we have learned in this course, and we'll discuss some topic also if needed. Okay. So I'm sharing my screen. Or obviously, if you have any question, you can ask. I think my screen is visible,
Yes, ma'am,
right?
it's visible.
Yes. So we'll do some MCQ type questions mostly. Okay, so the first question is, which of the following base defines a nanoparticle? Which of the following base defines a nanoparticle? So you can answer or you can write in a chat also. So everybody knows that nanoparticle is in the range between 1 to 100 nanometers. So any material which has a dimension between 1 to 100 nanometer is called nanoparticle. So this is option A. Next is which of the following is not a classification of nanomaterial based on dimension? Which of the following is not a classification of nanomaterial based on dimension? So you know about the 0D particles which have all the dimensions are in nanomaterial size. No, which of the following is not a classification of nanomaterial? So 0D is a classification of nanomaterial. So that is what I am saying. The 0D materials where all the dimension, that means height, width, and the length, all are in nano dimension in the range of nanometer. That is called 0D nanomaterials. Example like quantum dots. Right? Same. Similarly, one dimensional means two dimension is less than nanometer range, and one dimension is greater than nanometer range. You can remember the dimension of nanorod or nanowire. Okay, two dimension is one dimension is below nanometer in the nanometer range, and the other two dimension is above nanometer range, like nano sheet or graphene sheet, right? But there is another nanomaterial which is called 3D nanomaterial. There is no such called 4D nanomaterial. So obviously the answer will be 4D here, okay? Because in the question it is asked that which of the following is not a classification. So it is 4D. Okay. Next. Third question is of true or false type. So as the size of a material decreases to the nanoscale, its surface area to volume ratio increases, leading to enhanced chemical reactivity. True or false?
True.
Yes. So it is true. So I will refer to the class materials I have gone through. One. So you have seen this example of high surface to volume ratio, right? So when the side of the cube is 1 meter, the surface area is 6 meter square. When are we dividing into two similar parts? So the surface area is increasing to 12 meters square. When again this each cube is divided into two parts, the surface area is increasing like 24 meters square. So it is almost 4 times of the original one, right? So this leads to the answer that size of the nanomaterial decreases to the nanoscale, its surface area to volume ratio increases. So it is true. Question number 4. What is the main reason nanomaterials can exhibit different properties compared to their bulk counterparts? What is the main reason nanomaterials can exhibit different properties compared to their bulk counterparts? So it is also coming to the same reason that they have a large surface to volume ratio or large surface area, and quantum effects become significant, right? So this is option 4B. Next, which of the following is an application of nanoparticles in medicine? Which of the following is an application of nanoparticles in medicine? So in all the cases, we can use
Drug delivery.
nanoparticles. Yes. For medicine, it is the drug delivery. Next, which property of gecko feet enables them to adhere to surfaces and has inspired the development of advanced adhesives?
Capillary action.
Yes, it is also van der Waals action, because what happens, if you have seen the PDF of the gecko pad, see, gecko feet are covered with nano-sized hair. That means there are nanostructures at their feet, or whatever you can call it. There are nano-sized structures. So that nano-sized structures uses intermolecular forces. That is van der Waals forces. Here it is not the capillary forces, because van der Waals forces work in the operate in the nanoscale level. Okay, so that van der Waals forces means the attraction. Mostly van der Waals force defines the attraction between two materials. So it is that van der Waals force operating at that nanoscale level between the wall and the feet of the gecko is operating, and that is what that is there is that attraction force between these two materials, and that is allowing them to stick to the surface. So the answer is van der Waals forces due to nanoscale hair structures. Okay, is it clear?
Yes, ma'am.
Yes. Next is the lotus effect, which leads to superhydrophobic and self-cleaning surfaces, is primarily due to the presence of micro and nanoscale surface roughness combined with a hydrophobic coating. So it is also true, because you have seen yourself also that the raindrops cannot wet a lotus leaf because of this nanostructures on the leaf, and it also has a hydrophobic coating kind of thing. Okay, that makes lotus leaf very super hydrophobic. Next, which of the following is not a direct application of biomimicry in nanotechnology as described in the lecture? Which of the following is not a direct application? So, self-cleaning windows inspired by moth eyes, drag-reducing swimsuits inspired by shark skin, quantum dot solar cells inspired by butterfly wings, and magnetic resonance imaging inspired by peacock feathers.
MRI.
Yes,
Option D.
option D. So, MRI is not inspired by the peacock feathers. So this is not an example of biomimicry. Next, the iridescent color in peacock feathers and butterfly wings is caused by pigment molecules embedded in the nanostructure of the feathers and wings. Is it true or false? The iridescent color in peacock feathers and butterfly wings is caused by pigment molecules embedded in the nanostructure of the feathers and wings.
True.
No, how can? Because they don't have pigment. This is false. It is happening because of the layers, because of the nanostructure at the peacock feathers. So again, referring to the PDF or lecture, you can see that, see, this is the peacock feather. So when it is, we are zooming it, you can see the nanostructure. Again, you zoom it, you can see this nanophotonic crystals. Same goes for the butterfly wing. So what is the reason? Butterfly wings are layers of nanoparticles separated by layers of air. So there are several layers of multiple layers of nanostructure sheet, or in between those layers there are air, and that air thickness controls the color of that particular thing. So when this is nanostructure, so you know that if in nanostructures, when light is incident upon a nanostructure, it emits different colors because of the thickness of that particular layer. So that air gap between these layers control the thickness and also control the color we see. Also controls the color the light reflects and what we can see. So it is not the pigment, it is the nanostructure which is defining their colorful feathers. So it is, the answer is follow. So the right answer is structural color due to nanostructures or nano patterns within. Okay. Next, which of the following statements about the nanostructure of woodpecker beak is correct? The beak is solid and homogeneous, providing maximum hardness. The beak contains a spongy bone structure and layered keratin scales, which dissipate impact energy and inspire shock-absorbing materials. The beak is composed entirely of chitin, similar to insect exoskeletons. The beak's nanostructure is designed to maximize electrical conductivity. So what about this? Okay. So the structure at the nano level makes, so also these toucan beaks have, or woodpecker beaks have, these nanostructures, and this nanostructure makes these beaks incredibly light and strong. Okay, here. So woodpecker makes a blow into the tree and trunk. Its beak repeatedly strikes at a speed of 6 to 7 meters per second. The physical characters of the head include spongy bone on the upper back and extended hyoid bone, a tightly enclosed small brain within the skull, and a plate-like high-strength cranial bone. So these nanostructures make them, so this is not solid. So first option is wrong. This is also not composed entirely of chitin. And this big nanostructure is designed to maximize electrical conductivity. This is also not true. The answer will be, the beak contains a spongy bone structure because of the nano patterns and layered keratin scales, which dissipate impact energy and inspire shock absorbing material. That's why they can impact on the wood very fiercely, but still they don't get hurt by those movements, right? So it is option B. Next, mechanical milling is an example of which approach for nanoparticle synthesis? Mechanical milling is an example of which approach for nanoparticle synthesis? Yes, it is top down. So milling means it includes ball milling, rod milling, everything. Okay. So that means there is a big particles and we are hammering it and it becomes smaller particles, and thus these nanoparticles are synthesized. Okay. Next. In the laser ablation method, nanoparticles are generated by chemical reduction in solution, vaporizing a solid target with a pulsed laser, biological synthesis using bacteria, or electrochemical deposition. So in the laser ablation method, what was that we have seen in the first week of lecture? Yes, this one. This one. So this is the laser ablation method, right? So under solution, a target is given, a metal target is given, and this laser is incident upon this NDYAG laser is incident upon that specific target, and by vaporizing that target, it creates nanoparticle, right? So the answer will be vaporizing a solid target with pulsed laser. This is also another kind of top-down approach for nanoparticle synthesis. Next, the presence of a capping agent during chemical reduction prevents aggregation of nanoparticles and controls their size. So, what happens in the bottom-up approach of nanoparticle synthesis? So, generally, a chemical synthesis, if I talk about chemical synthesis of gold nanoparticles, so it is the gold salt is there, then sodium citrate is used as a reducing agent, right? Also, you can call it a capping agent, and then the nucleation happens, then the growth happens, coalescence happens, then this finally nanoparticle is formed. So, this capping agent are used for to prevent aggregation of the nanoparticles, right? So, this is true. This is true. Next question. Which phenomenon is responsible for the color of colloidal gold and silver nanoparticles? Which phenomenon is responsible for the color of colloidal gold and silver nanoparticles? So it is the surface plasmon resonance, right? What happens in surface plasmon resonance? So what happens? So when the nanoparticle size is very small compared to the incident electromagnetic wavelength, right? So what happens? The electrons in the conduction band of these nanoparticles are oscillating or starts oscillating upon the incidence of this electromagnetic wave, and when these two oscillation is in synchronization, then they produce a response that is called your surface plasmon resonance, right? So the lecture, if you see, so this is the surface plasmon resonance. So when a nanoparticle is much smaller than the wavelength of light, right? What is the incident light? That is what is the electromagnetic wave also. So this coherent oscillation means the oscillation of the incident wavelength of light and the electron is in synchronization. So this coherent oscillation of the conduction band electrons, these conduction band electrons are present in this nanoparticle induced by the interaction with an electromagnetic field. Here electromagnetic field is nothing but the light, right? So they show a resonance or color. So this resonance is called the surface plasmon resonance. So here the answer is this surface plasmon resonance is the reason for the color in this gold solution. So the answer is surface plasmon resonance. Next, homogeneous nucleation results in monodispersed nanoparticles, while heterogeneous nucleation leads to polydispersed particles. True or false? So what are these monodispersed and polydispersed particles? So you have seen earlier also that monodispersed particle means all the nanoparticles synthesized have that uniform shape. That is the monodispersed nanoparticles. And polydispersed means when they have different shapes, right? So what it is said, see. So in the nanoparticle synthesis process, first step is the nucleation. Nucleation means the first nanocrystal comes or first nanocrystal forms inside the solution. That is the nucleation. And that when this multiple nanocrystals are there, that is called the growth section, right? And now this next step is development of the facets. So depending upon the nucleation and growth, different shapes of gold nanoparticles or nanorods are formed. Okay. See. Okay, this is the nanorod. So here the first precursor is your gold solution. Then the nucleation. When the nucleation happens fast, then small nanoparticles are created or nanocrystals are created. When that growth happens, they create smaller nanoparticle size. When the nucleation happens very slowly, then limited number of nanocrystals are formed, and when the growth phase comes, they are agglomerated together and form bigger nanoparticles. Okay, so this is how we can vary the rate of nucleation to form the desired size of the nanoparticle. And see, homogeneous nucleation means all nuclei will be formed simultaneously. That means in the nucleation step, all the nanocrystals will form simultaneously, and that lead to similar size of nanoparticle. That is called monodispersed nanoparticle. And Heterogeneous means that nucleation of the different particles happens in different times, and that leads to different size or different shapes of nanoparticles. And this is called polydispersed nanoparticles. So as per this understanding, the uh, statement is true. Next, which enzyme is primarily responsible for the biological synthesis of silver nanoparticles in Bacillus licheniformis? Which enzyme is primarily responsible for the biological synthesis of silver nanoparticles? So, in the silver nanoparticle synthesis, you can see it here also. So, in the silver nanoparticle synthesis, silver salt will be required, right? Because in the case of gold nanoparticle synthesis, gold salt is required. That is why similarly, in case of silver nanoparticle synthesis, silver salt is required. That is, and here. So, in the silver nanoparticle synthesis, AgNO3 is the salt, that is silver nitrate. So, we need to reduce this silver nitrate, right? So, and what are the options here? You can see that nitrate reductase is here. So, we need to reduce this silver nitrate. Obviously, nitrate reductase will be the appropriate enzyme in case of biosynthesis. Okay, so the answer is nitrate reductase. Next question. Magnetotactic bacteria can orient and migrate along geomagnetic field lines due to the presence of magnetosomes. So from the statement itself, you can understand that this bacteria will follow the uh, global magnetic field lines, right? So this is your true. Next. Which of the following is not a typical advantage of biological methods for nanoparticle synthesis? Which one is the Which of the following is not a typical advantage of biological methods for nanoparticle synthesis?
Ease of controllable site.
The answer is B. Yes. High toxicity of byproducts. So you can control the size and shape in biological method also, depending on the time. Like if you keep that for longer time, it may form bigger size. But high toxicity byproducts, you cannot control that. So they will form. So that is the disadvantage also you can. Next is plant viruses are often preferred over human viruses for engineering viral nanoparticles because they minimize the risk of infection and immune response in humans. Yes, it is true. In viral nanotechnology, what is the main function of a protein cage architecture? to act as a reducing agent, to serve as a size constant reaction vessel and chemical building block, to increase the toxicity of nanoparticles to prevent nucleation. Yes, it is the B. To serve as a size constant reaction. Next, the redshift observed in the surface plasmon resonance peak of gold nanoparticle is primarily due to what? So SPR peak shifts because of the increase in particle size. So it is option B. Okay, so redshift happens. So when the UV characteristic curve you have seen, this is the wavelength and this is the absorbance. So this is from generally 200 to 800. It is the range is between 200. The UV visible spectra is between 200 to 800 nanometer. And when the redshift happens, that means it is going towards the longer wavelength. So it happens when the particle size increases. Okay. Next is DLS can provide direct information about the shape of the nanoparticle in solution. DLS can provide direct information about the shape of the nanoparticles in solution. Sorry for the network. Let me share the screen again. So where was I? The question was, DLS can provide direct information about the shape of nanoparticle in solution, which is a false. Which is false because DLS talks about your charges, not the shape of the nanopart. Okay. So which zeta potential value is generally considered the dividing line between stable and unstable colloidal dispersion? Which zeta potential value is generally considered the dividing line between stable and unstable colloidal solution? It is plus minus 30 millivolt. This plus minus 30 millivolt. So if you see this last first week lecture, so this is the from plus minus 30, this stability from plus minus 30 and above stability achieves, and below that plus minus 30 it is the instability portion, right? So the answer is plus minus 30. Next, in TAME, what is the main reason for using very thin specimens? In TAME, what is the main reason using very thin specimens? To prevent sample heating, to allow electrons to pass through and form an image, to increase surface area for interaction, to reduce sample contamination. What do you think? Yes, because in TAME what happens? The light in the image you can see here that so this is a TAME image. This is your TAME. So what happens in TAME, that electron beam transmits through the sample and creates the image, or image is captured in the detector, right? So to form an clear image, the sample specimen should be very thin. Generally, we use TAME grid for that. Okay. So this is option B. Next, atomic force microscopy can be used to image non-conducting biological samples in liquid environments without special sample preparations. Yes, this false, because liquid environment, you cannot do AFM in a liquid mode. You have, that has to be dry very well. So it is false. Next is, which of the following is a key driving force for molecular self-assembly in DNA nanotechnology? Which of the following is a key driving force for molecular self-assembly in DNA nanotechnology? Is it the covalent bonding? Is it the hydrogen bonding between complementary bases, metallic bonding, or radioactive decay? Yes, it is option B, hydrogen bonding between the complementary bases. Next, DNA origami relies on a long single-stranded DNA scaffold and many short staple strands to fold into complex nanostructure. DNA origami relies on a long single-stranded DNA scaffold and many short staple strands to fold into complex nanostructures. Is it true or false? Yes, it is true. Which property of DNA makes it especially suitable as a building material for nanotechnology? Which property of DNA makes it specially suitable as a building material for Nanotechnology. It is high electrical conductivity, its well understood base pairing rules and predictable structures, its ability to form metallic bonds, its resistance to enzymatic degradation. Its well understood base pairing rules. The stability of DNA nanostructures is unaffected by changes in pH or ionic strength of the solution. The stability of DNA nanostructures is unaffected by changes in pH or ionic strength of the solution. Yes, it is false because it is highly dependent on pH. Next, DNA origami nanopores can be engineered to selectively control the passage of which of the following? DNA origami nanopores can be engineered to selectively control the passage of which of the following? Only water molecules, ions, small molecules, biomolecules, proteins, or DNA strands. Yes, ions, small molecules, and biomolecules. Which structural feature allows peptide Lego molecules to self-assemble into nanofibers in aqueous solution? Alternating hydrophilic and hydrophobic amino acid forming beta sheets, random coil structure with only hydrophobic residues, alpha helix formation with charged side chains, or disulfide bond cross-linking between cysteines.
A. Option A.
Yes, it is option A. Next is lipid-like self-assembling peptides can form nanotubes and nanovesicles with diameters similar to those of biological phospholipids. Yes, it is also true. So before proceeding to the next question, so the thing is this call again will end in 15 minutes. I have to create another one and I'm sharing that link to the chat. Please join that. So I have shared the link. Please join there. So other people, please join here who have not joined the another call. Please join the new call, which I have shared. So I am joining the new call. Okay. So please join this. The joining time will end in 9 minutes. Okay. After that, this call will automatically end. Okay. So here I am sharing this thing. I think my screen is visible here. Okay. So let's start. Which of the following is a key advantage of using peptide nanofiber scaffolds like RADA 16-1 or Pura matrix for 3D cell culture? So what is the advantage of using this peptide nano fiber scaffold in a 3D cell culture? Yes, it is. They closely mimic the porosity and structure of natural extracellular matrices. Next, glyconanoparticles can be used to study specific carbohydrate interactions and have applications in anti-adhesive therapy against bacterial infections. So glycosanoparticles are nanoparticles coated with glucose or glycogen kind of carbohydrates, right? So it is true. So they are used for this carbohydrate interaction. So it is true. In confocal imaging studies, what is the main reason for the different localization patterns of acetylglucosamine encapsulated quantum dots and mannose encapsulated quantum dots on sperm cells? Yes, it is also the distribution of these two receptors on the sperm surface define this different location. Next is, which structural feature allows liposomes to carry both hydrophilic and hydrophobic drugs? Which structural feature of liposomes?
Option B,
Yes, this is the
bilayer
bilayer
structure with
structure.
hydrophilic core.
Yes, this is the bilayer structure of this hydrophilic core and uh, hydrophobic tail. Next is, stealth liposomes are designed to evade detection and clearance by the RES by incorporating PEG molecules on their surface. So these liposomes are designed to evade detection and clearance by the RES by incorporating PEG molecules on their surface. So these PEG molecules are hydrophilic, so that will help them to evade the detection. So it is true. Next is the which method is commonly used to prepare SUVs from MLVs. Which method is commonly used to prepare small unilamellar vesicles from multilamellar vesicles? The answer is sonication. So sonication helps to disperse in smaller particles. Okay, that is why large MLVs are forming smaller unilamellar vesicles in sonication. Next, solid lipid nanoparticles are more stable during storage and have less drug leakage compared to conventional liposomes. Solid lipid nanoparticles are more stable during storage and have less drug leakage compared to conventional liposomes.
True.
Yes, it is true. Nanostructured lipid carriers are considered superior to solid lipid nanoparticles for cosmetic applications because they are composed only of solid lipids. They have a perfect crystal lattice. Their imperfect matrix allows higher drug loading and long-term stability. They cannot encapsulate hydrophilic drugs.
Option C.
Yes, the imperfect matrix allows higher drug loading. Next, which molecular motor synthesizes ATP by using a proton gradient to drive rotation?
F1 ATPase.
Yes, the answer is F1 ATP. Next, the BZ transition in DNA nanomachines can be triggered by changing the ionic strength of the medium.
True.
Yes, it is true. In bacterial chemotaxis, what is the primary function of the chemoreceptors? Generate ATP, transmit information about attractants and repellents to the flagellar motor, synthesize proteins, or control DNA replication.
Option B.
Yes, this is the option B. Next, molecular orient communication between nanomachines can use calcium ions as information carriers in both direct and indirect access scenarios.
False.
It is true, I think, but I have to check once also. I will check it later, but first just complete. I think all this answers are given in the PDF, so we'll confirm there. Which of the following is a major challenge in designing autonomous bionanomachines for drug delivery? Which of the following is a major challenge in designing autonomous bionanomachines for drug delivery? So it is B, option B. Many-to-many communication and recycling of the carrier molecules. Which property of the carbon nanotubes is primarily responsible for their exceptional mechanical strength? So,
Option D.
yes, it is also the multi-layer nanostructure, also the hexagonal network. You have seen the nanostructure, carbon nanotubes are prepared kind of this, and they are long. So they have these hexagonal structures also. So this hexagonal network mainly gives the uh, mechanical strength. Next is the single-walled carbon nanotubes generally have fewer structural defects than multi-walled carbon nanotubes.
So the answer is B and D, the pre-46 question.
Actually, there are two type of nanotubes also, right? Single-walled and multi-walled. So multi-walled is multi-layered structure, you can say. But in single-walled, it is only a single layer. But the basic mechanical strength is provided by a hexagonal network. So I think it will be only one. Option B only. Okay. Single-walled carbon nanotubes. Let's find the carbon nanotubes first. So these carbon nanotubes are the sheet and that are rolled into a cylinder type. So this can have one or multiple layers. So when this is a single layer, that is called single walled carbon nanotube. When this is a multi-layer, this is called multiple layer. So but in single layered nanotube also have this hexagonal structure which give them the mechanical strength. So only the hexagonal one will be the correct one. Okay. Here, this is the single walled and this is a multi-walled. So what is the question? Here it is. Single-walled carbon nanotubes generally have fewer structural defects than multi-walled carbon nanotubes. So
True.
yes, it is true.
Oh, it's false, ma'am. The fifth point.
Yes, yes.
Chance of defect is
Chance
more.
of defect is more. So this is true. A chance of defect is more during functionalization and chance of defect is less, but once occurred is difficult to improve. Yes, it is false. Next. Which synthesis method for CNTs is most easily scalable for industrial production and allows control over tube length and purity? This is the chemical vapor deposition. Functionalization of CNTs improves their water solubility and biocompatibility, making them more suitable for biomedical applications. This is true. In CNT-based biosensors, what is the main advantage of using CNT tips over traditional microelectrodes? Increased background noise, lower chemical stability, maximum resolution, and high sensitivity due to small diameter and high aspect ratio, and reduced signal detection capability.
Option C.
Yes, it is option C. So if you have any question, you can also ask. I am just revising all the questions we have done or we have learned in this course. So if you have any particular question, you can also ask. So next question. Which of the following is a key advantage of using quantum dots over traditional organic dyes for cancer cell imaging? Lower brightness and photostability, ability to tune emission color by changing particle size, inability to be conjugated to biomolecules, poor water solubility. So it is the option B, because just like nanoparticles, quantum dots are also one kind of nanomaterials and their size is much smaller than the nanoparticles also. And depending on the size, their emission color is changed, and that is advantageous for the cancer cell imaging, and it can be done as per our requirement. This is the main advantage of quantum dots. Next, carbon dots are considered more biocompatible and less toxic than conventional heavy metal-based quantum dots for in vivo imaging applications. It is true, because
True.
it is an inorganic material, so it is more biocompatible, carbon material. In multiplex cancer diagnosis using quantum dots, what does each distinct emission peak in the fluorescence spectrum correspond to? In multiplex cancer diagnosis using quantum dots, what does each distinct emission peak in the fluorescence spectrum correspond to? The concentration of a specific organic dye, the presence of a specific biomarker or antigen, the number of cancer cells present, the amount of X-ray absorbed. So the specific emission related to the B, the presence of specific biomarker or antigen. Next, gold nanoparticles coated with PEG are used as CT contrast agents because they are rapidly cleared from the bloodstream by the RES.
True.
Yes, this. Next, which property of Gado nanotubes makes them highly effective as MRI contrast agents at low concentrations? The ability to emit visible,
Option D.
yes, high loading of the ions in heated nanotube structures. Which property of nanomaterials primarily enables enhanced drug delivery across cell membranes? So it is the very common one. It is the high surface to volume ratio. Because of
Option A.
it is the high surface to volume ratio, that's why they are able to go anywhere inside the cell. The use of targeting ligands or nanoparticles increases selective delivery to tumor cells.
True.
Yes, this is true. Which surface property modification is commonly used to increase nanoparticle circulation time in the bloodstream? Which
Digitization. Option B.
yes. Nanotoxicology studies have shown that smaller nanoparticles tend to be less toxic than larger ones.
False.
Yes,
False.
it is false. The main challenge in scaling up nanoparticle synthesis for clinical applications is maintaining batch-to-batch consistency, increasing particle size variability, increasing surface defects, lowering particle stability.
Option A.
Yes, because in every batch the particle size may change or particle shape may change because of the environment it is synthesized, right? Because the shape also depended on the temperature. Next, which of the following is a primary reason for using nanofibrous scaffolds in tissue engineering? They are always non-biodegradable. They closely mimic the scale and architecture of the ECM. They prevent cell attachment. They block nutrient diffusion.
Option B.
Yes, it is the ECM. Electrospinning allows for the production of continuous nanofibers with controlled diameter and alignment, which can be tailored for specific tissue engineering applications. It is true because in the electrospinning we can control the diameter also and we can control the alignment also depending on the particular voltage. Next, which technique is especially useful for generating scaffolds with specific pore sizes by adjusting polymer concentration and temperature? Which technique is especially useful for generating scaffolds with specific pore sizes by adjusting polymer concentration and temperature? It is the,
Melt spinning.
uh, I think it is thermally induced separation.
Oh,
The incorporation of magnetic nanoparticles into scaffolds can influence cell adhesion and mechanotransduction by increasing scaffold stiffness and providing micromotions at the cell-scaffold interface. It is true. In tissue engineering, which property of nanostructured hydroxyapatite is most critical for enhancing osteogenesis? So this one we can search. I'm not sure. Our nanostructure used to enhance osteogenesis, the formation of bone. So what is the question? In tissue engineering, which property of the nanostructure is most critical for enhancing osteogenesis? High electrical conductivity, nanoscale size and shape similar to native bone matrix, complete insolubility in biological fluids, ability to form random coils. So nanostructures used to enhance osteogenesis. So the form that leads to the nanoscale size, I think. So it will be option B. Nanoscale size and shape similar to native bone matrix will enhance the osteogenesis. Next, which of the following is a major advantage of using nano-sized artificial cells over micro-sized ones for intravenous delivery? Which of the following is major advantage of using nano-sized artificial cells over micro-sized ones for intravenous delivery? They are more likely to be filtered.
option B.
Option B.
B.
Yes. they can circulate freely through the smallest capillary. That is why you are using a nano-sized artificial cell. Pegylation of artificial cell membranes increases their circulation time in the bloodstream by reducing recognition and clearance by the immune system.
True.
Yes, this is which method is used to prevent immunorejection of encapsulated cells in artificial cell technology. Exposing cells directly to antibodies using ultra-thin artificial membranes to isolate cells from the immune system, increasing the size of the artificial cell and removing all enzymes from the cell.
Option
Yes,
B.
it is the ultra-thin. Modified hemoglobin-based artificial red blood cells do not require blood group matching and can be sterilized to remove infectious agents. This
True.
is true. In the context of synthetic minimal bacterial cells, what is the primary goal of genome minimization? To maximize the number of unknown genes, to define the minimal set of genetic functions essential for life under ideal laboratory conditions, to increase the cell's resistance to antibiotics, to make the cell as large as possible.
Option B.
Yes, it is the minimal set of genetic functions essential for life under ideal laboratory conditions. Which of the following is the major challenge in 3D bioprinting of solid organs like the kidney? Achieving vascularization and innervation, printing with only synthetic polymers, avoiding the use of any imaging data, using only 2D tissue construct.
Option A.
Yes, it is the vascularization. Selective laser sintering in medical 3D printing uses powdered material as the substrate and a laser to fuse the powder into solid structures. Selective laser sintering. Let's see.
True. I'm guessing it's true. True, ma'am.
Yes, I think also it is true. But yes, a printer uses powdered material as a substrate for printing new objects. A laser draws the shape of the object in the powder, fusing it together. So this is which property is most critical for a hydrogel to be used as a bioink in organ printing? High electrical conductivity, excellent biocompatibility and ability to support cell growth, complete insolubility in water, high melting point. It is the option B.
Option B.
Inkjet bioprinters are preferred for printing high viscosity bioinks and cell spheroids due to their ability to handle thick materials.
True.
Let's see one slide. The doses limited are regular to distribute tiny droplets of substrate according to the digital instruction. What is the question? These bioprinters are preferred for printing high viscosity bioinks and cell spheroids due to their ability to handle thick materials. Inkjet printers are mainly used in bioprinting for fast and large-scale product. One type of inkjet printer called drop-on-demand inkjet printer manufactures in exact amounts, minimizing cost and waste. Thermal inkjet printers actually heat the print head to produce air pressure pulses that force droplets from the nozzle, whereas acoustic printers use pulses formed by piezoelectric or ultrasound pressure. Viscosity based is not written anywhere, so I think it is true.
It's
Yes.
true, ma'am. It can handle.
In the context of nanotechnology-enabled organ printing, what is the main advantage of using genus-like tissue spheroids with magnetic nanoparticles? They are immune to temperature changes, they enable magnetic self-assembly and precise spatial organization during tissue fabrication. They dissolve rapidly in biological fluids, they prevent any cell differentiation.
Option
Yes.
B.
Which nanomaterial is most commonly used as a label in lateral flow assay due to its strong red color and easy functionalization? It is gold nanoparticles. Gold nanoparticles are used in lateral flow assay. Microfluidic paper analytical devices rely on capillary action to move fluids through patterned channels on paper. It is also true.
True.
Which of the following is a key advantage of point-of-care diagnostic tests compared to traditional laboratory-based tests? Require highly trained personnel, provide rapid results at the site of patient care, always require expensive equipment, only detect a single analyte. So it is the
Option
option B.
B.
In a lateral flow assay, the absorbent pad serves to wick the fluid through the membrane, increasing the sensitivity of the test. Yes.
True.
What is the main function of the conjugate pad in a lateral flow assay? To absorb excess sample, to store and release labeled antibodies or particles that bind the analyte, to immobilize captured antibodies, to provide a color reference.
Option B.
Yes. So if you see the image of this lateral flow assay, so yes, so this is the lateral flow assay. So this is the sample pack where the sample is given. This is the second one is the conjugation pack. Here the lines are like antibody and attaches to this line. Okay, so question is about the conjugation pack. This is the store third option. To store and release labeled antibodies or particles that bind the analyte. Which of the following is the key advantage of nanopharmacology over conventional drug delivery? Greater fluctuation in drug concentration level, ability to target drugs to specific organs or tissues, increased toxicity of all drugs, complete avoidance of pharmacokinetic interactions.
Option B.
Yes, it is ability to target drugs to specific organs. Cationic, which is option B. Cationic nanoparticles are more effective than anionic nanoparticles at fully diffusing throughout tumor tissue. It is true.
True.
Yes, because all our tissues are mostly negatively charged, so cationic nanoparticles travel more easily, diffuse more easily. Which approach is most likely to enable a drug to cross the blood-brain barrier for treatment of CNS disorder? Increasing the number of polar groups in the drug molecule using transporter mediated or receptor mediated delivery system, decreasing the drug's lipophilicity, administering the drug only as a suspension.
Option A.
Option A. Let me check. This is the blood-brain barrier. Relative impermeability of the blood-brain barrier results from tight junctions between capillary endothelial cells which are formed by cell adhesion molecules. Approximately 98% of the small molecules and nearly all large molecules such as recombinant proteins or gene-based medicines do not cross the BBB. To bypass the BBB and to deliver therapeutics into the brain, three different approaches are currently used: invasive, pharmacological, physiological. So, question was about which of the approach is most likely to enable a drug to cross the BBB for treatment. The number of polar groups in the drug molecule are using transporter mediated receptor mediated. Pharmacological approach consists of modifying through medicinal chemistry molecule that is known to be active against the CNS target to enable it to penetrate the BBB. Modification of drugs through a reduction in the relative number of polar groups increases the transfer of a drug. Reduction in the relative number of polar groups. Okay, so it is increasing. So here it is written that modification of drugs through a reduction in the relative number of polar groups increases the transfer of a drug across BBB. So here in the question it is given increasing the number of polar groups. So it is the opposite.
Yes,
So
ma'am.
it is option.
So it depends upon lipophilicity.
No, this is depend on transporter mediated. So it is written that consists of modifying through medicinal chemistry a molecule that is known to be active against the CNS target to enable and it to penetrate. So lipid carriers have been used for transport. That also can happen, right? Mm, I'm confused between B and C. Physiological approach is recognized by scientific community as one of the most likely chance of success. Transporter mediated receptor mediated. Which approach is most likely to draft to cross?
Given the ability of drug to come across blood-brain barriers primarily depend on its lipophilicity, not presence of polar samples.
Yes. So the answer will be C.
Yes, ma'am, it will be C.
Yes. It might be B also, but I'm not sure actually. I'm marking C for now. You can check. Next is controlled release nanopharmacology aims to deliver drugs at a predetermined rate, maintaining a constant therapeutic level over time. It is true. In cancer therapy, what is the main reason nanoparticles can accumulate more in tumor tissue than in healthy tissue? Tumor cells have smaller interstitial spaces Nanoparticles are actively pumped into tumors by healthy cells. Tumor tissue typically has larger interstitial spaces, allowing nanoparticle penetration and retention. Nanoparticles are only attracted to necrotic cells.
Option C.
Yes, this is the larger. Which of the following cellular uptake pathways is most likely to allow nanoparticles to avoid lysosomal degradation and deliver their cargo to the endoplasmic reticulum or nucleus?
Option B.
Yes, I think B or C both can happen because both of them avoid this lysosomal degradation. So it I am marking both. Okay, clustering mediated and caveoli mediated. The formation of a protein corona on the surface of a nanoparticles in a biological environment can alter their cellular uptake pathway and biological identity. The formation of a protein corona on the surface of nanoparticles in a biological environment can alter their cellular uptake pathway and biological identity, which is true.
True.
Which property of nanoparticles is most likely to increase their uptake by phagocytic cells such as macrophages? Small size, highly negative surface charge, opsonization by immunoglobulins and complement proteins, complete hydrophilicity. This is the option C. Cationic or positively charged nanoparticles can escape from endosomes after internalization due to the proton sponge effect, leading to perinuclear localization. Yes,
True.
that's true. Nanoparticles with which of the following surface modifications are most likely to exhibit reduced cellular uptake due to steric hindrance and hydration shell formation? Hydrophobic alkyl chains, polyphasic chains, cationic amine groups, anionic carboxyl groups. It is the PEG.
PEG.
Yes,
Option
it is the
B.
P. Which essay? Because PEG chains, that means PEG molecules are making a nanoparticle neutral. And it is not cationic or positively charged, and it is not making them negatively charged. So it is making them neutral. So that is why the cellular uptake will be reduced. Which assay is commonly used to determine the minimum inhibitory concentration and minimum bactericidal concentration of nanomaterials against bacteria? This is the turbidity optical density measurement. The disk diffusion method can be used to visually assess the antibacterial activity of nanomaterial incorporated composite nanofibers by measuring the zone of inhibition around the disk. Yes,
True.
it is true. Which staining technique is used to distinguish between live, early apoptotic, late apoptotic, and necrotic cells in anti-cancer studies of nanomaterials? So it is the AOED staining. A distinct DNA ladder pattern after agarose gel electrophoresis is a hallmark of apoptosis, while a DNA smear indicates necrosis. True or false? So apoptosis is the programmed cell death and necrosis is the sudden cell death, right? So DNA ladder is the hallmark of apoptosis. It is true. In flow cytometric analysis of apoptosis, which marker is commonly used to detect the externalization of phosphatidylserine on the cell membrane? Let's check. Let's check because not sure. Flow cytometry. So flow cytometry measures light scattering properties of cells, correlate to cell sizes and cell granularities, and fluorescence emissions of molecules attached to cells, usually conjugates of fluorochrome and antibodies to cell surface proteins. Qualitatively different concentrations of. So in the which marker is commonly used to detect the externalization of the cell membrane? I think it is B fix. Option B. Check this letter because I might be wrong, but what I think in our lab people do it with fixed, so that is what I am saying. Which property of nanomaterials most contributes to their increased reactivity and potential toxicity compared to bulk materials?
Option
Yes,
B.
it is that high surface to volume ratio that is the advantage for nanomaterial properties. Also, this is the disadvantage for this increased reactivity and toxicity. Inhaled nanoparticles can cross the blood-brain barrier and accumulate in neural tissue, potentially causing adverse effects. This is true.
True.
Yes. Which assay is commonly used to assess the genotoxicity of nanomaterials by detecting DNA strand breaks in mammalian cells? It is the COMET assay. MS test also does this, but it is not is detectable in, I think, mammalian cells. It is in different cells. Let me check. Yes, the MS assay detects neurotoxicity, DNA oxidative damage, but in Escherichia coli and other bacteria. But in eukaryotic cell or mammalian cell, it is detected by COMET assay. So the answer will be COMET assay. The hemocompatibility of nanomaterials is important for their use in blood contacting medical devices and can be improved by surface modification. Yes.
True.
In zebrafish embryo toxicity studies, what is a key indicator of nanoparticle-induced toxicity? Increased pigmentation, mortality rate, hatching delay, and abnormal phenotypes. Enhanced swimming speed, decreased water temperature. It is the mortality rate. Option B. Which of the following is not a consequence of increased surface energy in nanomaterial? Which of the following is not a consequence of increased surface energy in nanomaterials?
Reduced
Yes,
Gibbs energy.
it is reduced Gibbs. So, assertive statement is carbon nanotubes can behave as either conductors or semiconductors. The electrical properties of CNTs depend on the chirality and rolling axis of the graphene sheet. Which statement is true? So
Option A.
yes, both A and R are true, and R is the correct explanation. The COMET assay is used to detect DNA strand breaks and assess the genotoxicity of nanomaterials in mammalian cells. It is also true.
True.
Which method is not used to minimize the surface energy of nanomaterial?
Agglomeration.
Which method is not used to minimize the surface energy of the nanomaterials? In agglomeration, what happens? The particles come together because they don't repel each other anymore. But as per the system, that is the most stable condition. That is why these things are happening, right? So a system always tries to minimize its surface energy. So at that particular situation, I think agglomeration is happening because the nanoparticles are trying to minimize the surface energy. So it is not, it may not be the reason. The surface energy. But I think it knew.
That method is not
Yes,
used. That agglomeration method is not used, but it automatically forms for reducing surface energy, right?
yes. Agglomeration, yes. Agglomeration is not like expected. It should not happen. From that way you can see. Okay. It can be agglomeration.
Because if you agglomerate it, it won't be nanoparticle
Yes,
anymore.
no, no.
We
It
cannot
will
do that
settle
anymore.
down basically. And you can see the
Mm.
color change. Also, the surface plasmon property will be lost because it is settling down. Next is nanomaterials often exhibit quantum effects not seen in bulk materials. The reduced size leads to quantization of energy levels. So it is pretty clear. So it is the two
True.
option.
Option A.
Which method is irreversible for reducing the surface energy of nanomaterials? It is also agglomeration. Yes.
Agglomeration.
The rolling axis of a graphene sheet determines whether a carbon nanotube behaves as a conductor or semiconductor. Yes,
True.
it is true.
Ma'am, I think you missed one slide
Okay.
37.
Oh, yes. Nanomaterials have a large fraction of surface atoms compared to bulk materials. This leads to higher reactivity and unique catalytic properties. True. Yes.
True.
Option.
Option A.
Which property of nanoparticles provides a driving force for diffusion? It is also the high surface to volume ratio.
Option B.
Which of the following is not a benefit of the bottom-up approach in nanomaterial synthesis? Less crystal defects, homogeneous chemical composition, better short and long range ordering, less surface energy.
Option C.
Option C, better short and long range ordering. Yes. Nanomaterials are highly reactive compared to bulk materials and they have a large fraction of surface atoms. This is also obviously true. So the question we have done.
Option
Which property of nanomaterials is most responsible for their unique catalytic activity? This is the high surface to volume ratio. In
Option
heterogeneous nucleation, introducing a new surface changes the surface energy but does not affect the Gibbs-free energy if precursor concentration and temperature are fixed. True.
True.
What should be the contact angle between substrate and growth species for a zero critical energy barrier in heterogeneous nucleation?
45 degrees.
Yes. If the Gibbs-free energy of homogeneous nucleation is 10 kilojoule and the contact angle is 30 degrees, what will be the Gibbs-free energy of heterogeneous nucleation? The answer is given 0.1 kilojoule, but I am not sure from where it is coming. So you check please. To obtain monodisperse nanoparticles, what should be the sequence of nucleation and growth of the precursor? First nucleation, then growth, and nucleation discontinue when growth starts. First growth, then nucleation is not true. Both nucleation and growth start at the same time. First nucleation, then growth, and nucleation continues when growth happens. It is option A. Nanomaterials have reduced imperfections compared to bulk material due to their small size. It is true.
True.
Which of the following statements about carbon nanotubes is correct? CNTs are always metallic. CNTs are made from a single sheet of graphite rolled into a tube. CNTs have only one,
Option
yes,
B.
CNTs have only one configuration. CNTs cannot be used in composites. So it is option B. Nanomaterials are used in fuel cells, catalysis, and sensors. Their high surface area and reactivity make them suitable for these applications. So this is option A.
Option B, ma'am. R is not correct explanation, right? It does not react.
Oh, does
Nanoparticles
not
does
react.
not
Okay.
react.
Sorry. Thank you. So option B. Next, which property of nanomaterials is most important for their use in drug delivery systems? This is the high surface to volume ratio.
Option.
Yes, so these are the questions. Because this type of MCQ questions, I think, will be coming in your exam if you are giving the exam. So that is why I have going through all the question answers or MCQ. Obviously, go through the lectures carefully because that is what is important. And also in the second week lecture, there is so many types of particles or phospholipids are there, which is very confusing sometimes. There are solid nanoparticles, nanostructured lipid particles. There are different types of liposomes. So remember those differences, otherwise it will be confused in exam hall. So I just want to go through that portion once. So glyconanoparticles is the nanoparticles are when they are coated with any type of carbohydrates, right? So this is your basic lipid. So it has a hydrophilic head, which has phosphate group and glycerol chain, and it has hydrophobic tail, right? These are the saturated fatty acids. And when this has two hydrophobic chains, these are called phospholipid. When they have three hydrophobic chains, they are called triglycerides. Okay. So liposome is the double layer of this type of chains, right? So you can see here. So here you can see that these bilayers of phospholipids are self-assembled to form a liposome. So there are different types of liposomes used in used in drug delivery or many things. So you can say hydrophobic drugs are delivered, hydrophilic drugs are delivered, positively charged lipid, negatively charged lipid. This is the conventional liposome. Uh, these liposomes can be functionalized with target ligand, and that can be used in theranostic liposomes, right? These ligand target liposomes are functionalized with small molecules, analyte, carbohydrate, antibodies, protein for that particular reaction. And liposomes can also be pegylated, that means neutrally charged, okay, for some other reaction. And these liposomes can be divided into various types of life vesicles depending on their size and self-assembly. So when the size is small, that is called SUV, small unilamellar vesicles. Second one is the large unilamellar vesicles. Third one is the giant unilamellar vesicles when the size is greater than 1000 nanometer. Okay. So when these layers are in two forms, and in between these two layers there is water. This is called the oligolamellar vesicle. This is the multilamellar vesicle. That means multi layers of this bilayer compositions are there. Okay, and when this is the different size of multilamellar vesicles are there inside a bilayer system of lipid, this is called multivesicular liposomes. Next is the so this is the different types of phospholipids. So this is This is the micelles. When these lipids, or when these lipids are just arranged with their hydrophobic, one hydrophobic head and one hydrophilic head and one hydrophobic tail. When that is formed in around the particle, that is called the emulsion. When there is a lipid particle inside them, that is called solid particle inside them, that is called solid lipid nanoparticles. Okay, when this there are structured solid particles inside the core, this is called nanostructured lipid particles. This is liposomes, and there are other types of this structure. So here this is one. So this is the liposomes, and this is the micellar formation. Okay. So how these sizes depends on the formation of different types of self-organized, and all these structures are self-organized. So they are not forcefully synthesized. They are self organized or self-assembled. So depending on the size of this hydrophilic head and hydrophobic tail, different types of self-assembled structures are formed, which are used in different applications. Okay. So this part is very important. Also, this DNA, there are different types of DNA, DNA, B DNA, Z DNA, how they are formed. All these things are discussed in the respective classes in the previous weeks. You can go through the lectures uh, uh, from YouTube. Also, the study material is already shared with you. I hope you have found it. And there are important YouTube links are given there to refer to your knowledge. These are the carbon sheet, the graphene sheet. And this is how carbon nanotubes are formed. Okay, in the third week, this is the last week, right? Yes. Third week, we have studied about this organ, bioprinting and other organs, right? So this is the difference between benign tumor and malignant tumor. Then comes the application of quantum dots. Then comes the synthesis of nanofiber through electrospinning. So you can see that depending upon the voltage, this uh, polymer solution is coming out, and depending upon the high voltage, these nanofibers are formed. Okay. So this is the organ printing, and there are reference of some papers also how people have used this bioprinter for the formation of bioscaffold. Okay, or 3D organ printing, how people have used this. So you can refer to these papers, you can go through these journals to refer at how these microfluidic based 3D scaffolds are formed in bioprinting artificial printers. Okay. And in the last class, we have learned about the point of care testing devices, why these are very much needed, because these things does not need any conventional method, any expert to test. It is affordable, sensitive, specific, user friendly. Anybody can use. You don't need very scientific knowledge to use these devices. Okay. And these are very and most importantly, these are portable, so it will be very much helpful for the uh, last mile population, right? So this is, and these point of care devices can also be some Paper based also, just normal paper we use in daily. So these paper based devices are very much, very much helpful to to fabricate a simple system for testing. Okay. So this is your LFA, lateral flow assay. This is the same. This is the sample pad. This is the conjugation pad. This is the reaction pad, and this is the absorption pad. There are four pads. Our pregnancy kit is based on this LFA conception. Okay. This is a microfluidic paper-based array where different analyte is given to the different portion, and when the reagents is given, it is detecting in different getting in different ways. Right? Our pulse oximeter is also a portable device or also a point of care device. Okay. So these are the different types of device is fabricated in our lab. So that is for this is for the lipase detection. You can go through the papers from the details from the reference. This is for the amylase detection. This is for uh, urinary tract infections detection. This is for the glucose beta microglobulin detection. This is for LEDR based paper based amylase detection methods. This is for all type of blood parameter detection, and this is for hemoglobin detection. So then comes the different types of endocytosis, that means cell uptake, phagocytosis, that is cell eating, and pinocytosis, that is cell drinking. In pinocytosis, that is also divided macropinocytosis, clathrin dependent pinocytosis, caveoli dependent pinocytosis, and clathrin and caveoli independent pinocytosis. Okay. So this is the different types of endocytosis. You can see how much it is dependent on the size of the cell or size of the nanoparticles. Okay. So this is the phagocytosis process. You can go through the lectures and learn. So this is the smaller solute for pinocytosis. This is clathrin mediated in endocytosis. This is caveoli mediated endocytosis, where a special caveoli membrane or clathrin membrane are formed around the nanoparticle or nanomaterial. This is your macropinocytosis. So this is the difference you can see here. Next comes the apoptosis and necrosis. So apoptosis is the programmed cell death and necrosis is the cell death after injury. So here you can see a normal cell goes for apoptosis. This is cell swelling happens, but in case of apoptosis, no cell kind of the cell swelling are there. Okay, so what happens? This cell is fractured and forms cell debris. But in case of necrosis, this cell swells and all the extracellular matrix are coming out of the cell, and it leads to cell disruption. And now, after some time, this all the cellular matrix are out in the blood, and all the cells are out also in the blood, and that leads to inflammatory response in your body. That means that cell will not survive anymore. Okay, but in case of apoptosis, this cell fragments, but cell debris are formed, but they are not dying. They are forming different apoptotic bodies, and these different apoptotic bodies are engulfed or swallowed by other phagocytes, other phagocytes. So that is not lead to complete death of the cell. Okay. So this is the basic difference between apoptosis and necrosis. So you can just see here, apoptosis is a programmed cell death, an organized energy-dependent process where cells systematically dismantle themselves in a controlled manner to remove unneeded, damaged, or abnormal cells without causing harm to surrounding tissue. So it is good. This type of cell death is good for your body. It is essential for proper development and maintaining the balance of cells in the body, contrasting with necrosis, which is uncontrolled cell death from injury that causes inflammation. So the four stages of apoptosis are initiation, when a signal triggers the process, the early or mid stage involving caspase activation, cell shrinkage, and chromation condensation. That late stage characterized by membrane bleeding, formation of apoptotic bodies, and DNA fragmentation, and clearance or phagocytosis, where macrophages engulf the apoptotic body. And in case of necrosis, is the irreversible cell death. Okay, so this is not good for your health. This comes after some high injury. Okay, tissues in a living organism typically caused by lack of blood supply, infection, injury, or toxin rather than programmed cell death. It is an uncontrolled process. So in apoptosis, you have control over the cell death, but in necrosis, you cannot have any control. uh, Swelling rupture, releasing its constituents into surrounding tissue and often triggering an inflammatory response. Okay, this leads to pain, swelling, visible skin changes, and other things. So this is the basic difference between apoptosis and necrosis. Okay, so I think this is for today. I hope these lectures, all the five lectures, will are a little bit helping you to understand the course biomedical nanotechnology. And if you still have any further question, you can write definitely. And if you want to go through the lectures, it is available on YouTube, and also the study materials are available in Google Drive. So you can refer anytime. And obviously, the lecture provided by professor is very important for the exam, because the exam will be completely dependent on the lectures he has given. So go through those things very carefully, and also refer to the videos, the YouTube videos I have shared for your help. Okay, so all the best for your exam, and we'll probably meet in next semester with other courses. So till then, goodbye.
Thank you.
Thank you.